you, Chairman. And so the title of my second talk is Modeling Detection of Key Biomolecules with Enzymatic Electrodes, Diffusion towards Randomly Distributed Active Sites. And here I would like to consider some theoretical aspects of the detection with the enzymatic electrode. Okay, here I put, for an example, the same electrode which was used in the previous talk, but basically this will apply to any other enzymatic electrodes or any other systems which build the same way. The point is here is that the enzymes which are distributed at the electrode surface, basically we cannot control precisely the position on the electrode surface. And if we look to kind of model representation of this electrode, the enzyme will be randomly distributed along the electrode surface. Okay, and the substrate have to diffuse toward the electrode surface and then find the particular active sites which will be able to process the substrate molecule. And this situation is not unique to the enzymatic electrodes. Okay, there are many other systems in natural sciences where this kind of interfaces are present, and in particular in physical chemistry. Okay, there are numerous examples, like for the nanoparticles, okay, in the inner substrate, the catalytic materials. It could be also the pores in the polycarbonate membrane, including the cases where the active center is located within the pore, okay, or it can be any defects in the monolayer or any other layer coating on the electrode. Okay, and in the following, I will consider as an archetypical situation this one, okay, where we have the nanoparticles distributed on the flat substrate, because if we will understand all the complex and intrinsic features of this system, it will be rather straightforward to extend it to the cylindrical nanoelectrodes. And the main question which I would like to address here is that, okay, we have the randomly distributed active sites, okay, and if there are any consequences on the mass transport and hence on the current which we measure in the experiment. Okay, and in the following we will consider these three assumptions. Okay, so we assume that the simple electron transfer occurring at the surface of the active sites, that the substrate is completely inactive, and also that the sizes of the active sites is more or less the same. And to speak about the effects, okay, I would like to remind you what we know from the theory of the regular arrays, that basically there are three important regimes. So that at the very short times we have a kind of planar diffusion wave just above the electrode. At the intermediate times, if the separation between the electrode is enough, we have a kind of diffusion spherical level. And at large times we have, when all the diffusion layers are overlapping, we have a single planar diffusion towards the electrode, toward the substrate surface. Okay, and evidently in this case, since we have just the planar diffusion, the mass transport will be relatively simple in the bulk solution. But what we want to understand is how is the current density distributed among the active sites, okay? Since here their position is not ordered, so this means that depending on the local position, they will act very differently, okay, in terms of the conversion of the material. And just to show you the approach, okay, how we can address this kind of system, we use basically the feature which is used also for the regular arrays, where because of the symmetry, one can identify a single unit cell and basically model all the process in this single unit cell. And just by multiplication to the number of the unit cell, one obtains the total response of the system. And we cannot do it straightforwardly for the system with the randomly distributed active arrays, but we can kind of generalize it, this approach, and use the Voronoi tessellation to construct this irregular unit cell, which basically represent for each of the unit cell, we regroup all the points which is the closest to a given active site. Okay, and then by projecting these kind of polygons into the solution, we get the unit cell for this complicated case. And in the same way, the total current will be summation of the current in of each of the individual cells. The only thing that each of the individual cells, they are very, very different, okay? In terms of the shape, in terms of the location of the active sites in it. So what we do, we again use the similar methodology as used for the modeling of regular arrays, and we basically uniformize or standardize the unit cells by converting them into the cylindrical unit cells with the centered electrode. And this allows us to write down more convenient form where we have just the response of a given cell with a given dimensionless surface area, which we define as the ratio between the unit cell surface area with respect to the surface area of the active site. And then we simply weight this kind of responses by the presence of the unit cell with a given surface area. And this is already a very efficient formulation which allows us to model very quickly this kind of behavior of this kind of system. And without going into the details, I just would like to say that here, basically in order to compute the response of this of the random array, we need to supply two components, okay? And this one we can evaluate once and for all, and we can do it even analytically. And the only things which is missing is the distribution of the surface area in the array, which will be depending on the particular system we study, okay? But as soon as we can introduce this distribution, okay, we can compute the responses for very different systems, for the regular arrays, for the random arrays, or for the random arrays with a given bias, as for example shown here, where the active sites have tendency to be located in the center of the array. And in addition to that, we can go in a reverse direction and basically try to extract this kind of information from the experimental current. So in principle, we show that this is possible. Here you can see the correspondence between the actual distributions and the our reconstructions. And okay, this is just to show how we can treat this system. But coming back to the question which I asked at the beginning, that if we evaluate, okay, how the dependence between the position of the active site and the current density, okay, which passing through it. And if we consider, for example, this kind of infinite periodic array, okay, we can identify a single cell which looks with these three active sites. And what we want just to compare is how the current densities are distributed for this kind of system. And we would like to compare with our model representation, which I showed you before, based on the Voronoi tessellation, where each of the active sites is located into the Voronoi unit cell. So basically, there is no interaction between the electrodes in this case. However, in the first case, the electrodes can interact diffusionally freely. Okay, and if we compute the amperometric current for these two systems, we see that the total current, which is given by the red curve, basically if we superimpose them together, so they are superimposed and the error is less than the 4%, which is perfectly fine for this kind of models. However, if we look to the individual contributions, that the currents at each active sites, we see that in two cases they are very, very different, okay? And to understand that, we can plot the flux lines, okay, originating from each of the active sites. And what we basically see here is that the active sites with the smaller Voronoi unit cell is basically invading into the volume of the active sites with the larger Voronoi cell. Okay, and if we make the kind of cross-section at different heights, okay, and measure this kind of distribution between the active sites and make a kind of Z stacking, we see this kind of picture. We show us that, in fact, actual distribution is quite different from those which are predicted from the Voronoi tessellation. And we also show that this effect is really kinetic dependent. So, for example, for the slow kinetics, we see that the distortion is much higher than for the case of the fast kinetics. But the most interesting part, if you look to these pictures quantitatively, in particular, if we consider the slow kinetic case, we see that the Voronoi surface area are given like that. Okay, so basically we have quite significant difference between each active site. However, for the for the unit cell, which are delimited by the flux lines, we see that basically they are the same, and this means that the system tried to redistribute equally between the active sites. Okay, and for the fast kinetics, we see that the numbers are not really equal, but the tendency is exactly the same. So we have a kind of equilibration between the unit cells of the surface between the surface areas of the unit cells in order to be more or less equally distributed. Okay, and what this basically gives us that when we plot the distribution of the active of the surface area of the active site, so we have this kind of log normal distribution given by the red curve, but due to this diffusion interaction, the differences between the unit cells is significantly decreased, and what we obtain actually is something which is basically a kind of Gaussian and is much narrow in its width. Okay, so basically tending to the same unit cell. So what we conclude here is even if the active sites they distribute randomly, okay, and the unit cell have sophisticated shapes, however, the cross section tend to be more or less the same, and this value to which they tend is basically represented by the average value of the found in the array for each of the active site. And this basically allows us also to explain why the early theory of the random arrays were quantitatively successful, okay, which this seminal paper was published almost 40 years ago, okay, by Professor Amatore. And in this work, it was considered the partially block electrodes, okay, when the electrode during the measurement is full, okay, or biofull. And each time for the this kind of random system, what they put into the correspondence the array with the hexagonal structure, and doing it in such a way that the surface area of the unit cell is equal really to the average surface area in the whole array. Okay, and so basically this is exactly what we observed in our more detailed simulations, that we have a kind of uniformization between the unit sides. So finally, what I would like to conclude is that with this kind of approaches, we can model random arrays with very different distributions of the active sites. Okay, we just need to provide the distribution of the element of the surface area of each unit cell. Okay, and we basically, in principle, it's possible to characterize this kind of systems statistically by extracting this information from the electrochemical signature of the system. And also we developed a kind of easy way to quantify this diffusion interaction between the active sites. And the most important one is that the diffusion interaction tend in such a way to reduce the difference between the active cells in the random array. And this means that the random arrays appear as not as random and actually quite uniform, okay? And this evidently allow us to facilitate in the future analysis for the measurement done on the enzymatic electrodes. Finally, I would like to thank my senior colleagues, okay, Professor Cristian Amatore and Professor Svir, and also younger colleagues who were involved at different stages of this work, Dr. Sliusarenko and Dr. Giovanni Pireddu. all these agencies for the partial support, and also I would like to thank CNRS Institute of Chemistry for the emergency at ENC grant, which support part of this work. And again, thank you for your attention.